It's a very great pleasure for me to introduce to you Dr. Gerhard Kasper, your keynote speaker today.
Thank you very much for this kind introduction, and I would also like to express my gratitude to you, to the audience, for giving me a chance to talk about some of the issues that were just addressed a minute ago. When I was invited to give a lecture here, I was thinking about what to talk about. And there will be various papers here at this conference about identifying sources, about characterizing emission mechanisms and methods also for characterizing it. In fact, within your conference papers that you've received, there's an excellent summary by Thomas Kuhlbusch and colleagues about what is known today about workplace measurements. And so I thought that it would be more interesting perhaps to take a step back and to ask some of the more, let's say, general questions about workplace aerosols and workplace aerosol measurements also, and which are phrased here in my title slide. What do we need to know about them? With what accuracy do we need to know? What would we like to know about them? And how do we go about finding out? I'm going to do that with a fairly broad brush. And when using a broad brush, one risks generalizations, and I hope I won't offend anybody by generalizing and by simplifying and leaving out some of the details. But I would like to start with a slide that I showed essentially recently at a review meeting of an EU project called NanoDevice, and which I think would be a good starting point here today. If you look at the life cycle or the pathway of the decision-making process that we are taking from an emission to a risk assessment, we begin at the source with the emissions. And then from that source, the aerosol travels somehow to the receptor, to the human receptor. And for an aerosol person, which I am essentially, the receptor is pretty much the entrance to the human airways, if we are speaking about humans in this particular case, or let's say the surface of a biological system if you think of a plant. From there on, you compute a dose, which is essentially the combination of a certain exposure time and an exposure concentration, if you will, plus you have to fold into that an efficiency for airways deposition, which is also an aerosol problem, if you will, and there's a lot known about where and how particles deposit in the human lung and the human airways, not just the lung. And again, this is very much particle size dependent, so particle size obviously is an issue that we need to know something about in order to get accurate dose information. And from there on, you proceed to establish those response relationships, the effects. And many of you do that here in this audience, and with great expertise and skill in designing experiments to tease those kinds of informations out of a system. And once you understand those, and once you have a measure, I should say, of those, and when you have a measure of effect, you can then also estimate risk. So in fact, you can minimize risk, or you can control risk via the effect. You can just minimize that, and for that you need some dose information. So in other words, people who do exposure studies and people who then therefore are able to compute a dose can provide for some information for that. On the other hand, the people who study the effects will also have to provide some information to the people who study exposure and who calculate doses, because you can of course also control the risk via the exposure, but you also need to know what metric to look for. In other words, you have to understand how particles interact and what aspect of a particle interacts with, say, in generally a biological system. So let me start by focusing on the effect side of the things first, and sort of try to sum up in a slide or two what I think we know about those response relationships. So what is known about the effects, about the bioresponse to particles? Well, without even knowing a lot about this discipline, one can fairly easily say that particle-related factors will be their concentration, their size, obviously. We wouldn't be talking about nano here in this audience if the size effect were not a critical aspect of this particulate nature. And of course, we need to know something, some minimum amount of information about the chemical composition. We need to know what kind of particles, what material we're dealing with. Of course, there are already complicating factors there, and we've already heard them today in some presentations. The surface or the surface state of a particle also plays a role with adsorption of species and what have you not. But I'm going to try and leave that out for the moment. So this is the minimum information. There's another aspect to a particle, and that is it has structure. And let's leave out fibrous materials. Let's leave out carbon nanotubes for the moment, because this is obviously an issue there, that structure, size, shape, it's absolutely not spherical. But even the materials which are more or less isometric in terms of particle size, there's an issue with structure. And the complication is that the particle is not a molecule. Molecular species are very easy to deal with in terms of any kind of dose-effect relationship, because all you have to do and all you need to know is their concentration in the fluid, assuming it's uniformly distributed inside. Particles are not that simple. What constitutes a particle? What is size, therefore? And if you look at the picture to the right, which I'm using a titanium dioxide particle there kind of metaphorically, this, to an aerosol scientist, was one particle. In other words, it moved through time and space as one entity. So for me, the red brackets here, this is something that's related to size. Not to structure, because the structure is more complicated to describe, but at least to size. If many people handle nanoparticulate materials in the form of a powder, and the starting, or a suspension, that's already better, but a powder. You have a starting point of some kind of an assay is a powder. Okay? So what is the particle size in that powder related to the transport properties of these particles in air in the aerosol form? In fact, what you will find very often is that, especially when electron microscopic information is available, is what an aerosol scientist would call the primary particle. The primary building block of this particle, where the red arrow points to, that's the particle size. And in fact, it's relatively easy to measure this particle size when you have a powder, because all you have to do is measure the BET surface area. And if you have a gram or so of that, that's something you can readily do. And from the BET surface area, you can calculate a particle size. But that's not the particle size which is transported through the air. And I would venture to say that that is not really the particle size which is responsible for the mobility of the particle as it moves through an organism, because the mobility also has something to do with size and shape. And let us assume that this titanium dioxide agglomerate here is a unity and stays integrated as one piece. Then that's what travels, not the small size. Okay. So here you already have a differentiation between a size which is responsible perhaps for an effect and a size which is responsible for mobility. So this does impose some difficulties, conceptual difficulties also, and it also has some implications, of course, for people who try to characterize materials so that in the end you can do a dose assessment or a risk assessment. Another very interesting aspect, which is not at all addressed at the moment, to my knowledge, is what structural features of an aerosol particle, of a complex structured aerosol particle, are preserved when it moves from the aerosol state to the, let me call it, the physiologically embedded state, when it moves into cellular fluids or extracellular fluids. As you know, surface tension does all kinds of things to particulate structures, and therefore that is a really unaddressed question. And so ultimately you could ask, what guidance can be given to the exposure side for measuring the right kinds of things on nanoparticles, engineered nanoparticles, if you will? Well, we do know, and to my knowledge at least, we do know that the effects are mass concentration related, but there's an increasing amount of evidence that there's also a surface proportionality in effects. And so clearly these two metrics are metrics which we should be addressing. A question which I've asked myself while preparing that slide is, how well are nanoparticle dose-response relationships established today? For a chemical engineer, as I am, I'd be drawing a line and saying, well, I have a response, and then I have varying concentrations, and I have various gradients from, let's say, between the inside and the outside of a cell, and therefore I get a dose-response relationship. I realize that these are much harder to get in such multi-parameter systems as you're looking at, but would it be fair to say that there's order of magnitude type of knowledge available today, that if you change your concentration, let's say by an order of magnitude, you could tell whether you were somewhere at the threshold, at the PEC concentration or not concentration, as we've heard this morning? I would say that's maybe a fair assessment. And that will help me to sort of deduce from that some requirements for accuracy also on the measurement side. Okay? Because the measurements are, of course, also not arbitrarily accurate, as you can imagine. And just dwell a little bit on this particulate nature. What I'm doing here is I have a sort of a backbone of my presentation, then I'm sort of interspersing some slides from my own field of work as an illustration for some of the issues. Some years ago, we've developed a technique for fragmenting nanoagglomerates with a device which is essentially an impactor where we can control the impaction velocity of the particles over a very wide range. And what you can do is for different materials, and these are all inorganic, silver, nickel, titanium dioxide, if you increase the impaction velocity, let's say, you can increase the degree of fragmentation. You can measure this fragmentation curve. You can see that some materials will break up with reasonable degrees of energy into essentially their individual constituents. The nickel here won't break up at all, for example, though it's very much in the same size range of a few nanometers. And some other materials will break up into subconstituents, which are not necessarily the primary particles, because the primary structures of this titanium agglomerate here were really sintered due to the production process sufficiently to never come apart again. But let's say if this was the aerosol particle that would have entered the lung fluid, and I'm now trying to build a bridge, would it then in the tissue fluids have perhaps broken up into these constituents and not any further? And would those actually be the mobile subunits which you should be looking for when it comes, for example, to franchising the boundary between across a cell boundary or the likes? We've also found similar things with diesel soot, and this is actually a fragmentation degree curve here with and without adsorbates on it, and you can see that actually you do not reach 100% defragmentation for diesel soot either. In fact, without adsorbates you get to about 80%, and I apologize for the bad pictures, but I couldn't in the short notice come up with any better ones. But what remains also for the diesel soot is substructures which are no longer broken up. I've also looked at the aspect of what we need to know from the, let's say, from the regulatory side. And what I find, and please correct me if I'm wrong, I find that we are dealing mostly with total concentrations as thresholds, mass, number. The structural or shape information is not really entering very much, except for some very obvious and specific situations, such as, for example, asbestos. I don't know how far we are along with CNTs in this regard, or whether there are already any kind of structural requirements there. And as you know, and we've already seen it, CNTs are very complex. They can be anything from individual fibrous materials to essentially spaghetti, which are complete, and it becomes very difficult to do that. Okay. And when it comes to concentration, particles are really treated more or less like molecules. Okay? The structural aspect of the particles is not considered. On the other hand, if you look at lung dosimetry, the basis of calculating a dose in your lung, you do need size information. You need aerodynamic size information, because the size distribution is the basis for calculating a deposition rate and also deposition location. So you really can't do without the size information, some size information about the particles. Let's now look at the other side of the issue, the exposure side. This is where aerosol measurements come into the picture. You need information about concentration, size, structure. Those are the minimum requirements, I would say, that you need for particles. What information can aerosol measurements provide for the workplace in real time and with what accuracy? I've highlighted the words for the workplace because those of you in this room who do aerosol measurements know that one can do fantastic things with aerosol measurements. One can work with great precision. One can tease out a great deal of information from these systems, but not necessarily in the workplace conditions. So I'm going to limit this discussion. And I've also added real time. That was my choice. Of course, there are techniques which are based on sampling the particles and then going to an electron microscope or doing some kind of chemical analysis to look at the speciation of the materials. But I'm going to leave that out because we live in an area where we'd like to know things as quickly as possible, which means right away. Aerosol measurements can provide size distribution information by number or surface area by direct measurement. In other words, you have devices which will give you this information sort of as the direct information. You do realize that particle size or diameter is an operational definition in this context. When you measure particles with any kind of device, you don't really measure size physically like you would do in an electron microscope. You measure mobility, aerodynamic behavior, optical behavior, and so on and so forth, and therefore you always have an equivalent size. These equivalent sizes don't perfectly agree with each other. Of course, there is a science that connects them to each other. There are conversion factors which you can use, shape factors and have you not. But that already assumes that you know a lot about the particles. And that's typically not what you have when you look at the mixture of aerosols in the workplace. So the comparative uncertainty between the different techniques, if lab A or workplace A uses this device and B uses another device and C again a third device, is something better than a factor of 2 in size, which isn't bad. If you know particle size, let's say, well, it's not 100 nanometers, but it's maybe 200 nanometers, that's not bad, this kind of information. Of course, a factor of 2 in size implies 2 to the second power for the surface area and 2 to the third power, which is 8 in volume uncertainty. So you see we're not all that far away from the factor of 10 in concentration, because volume essentially is concentration that I'm also surmising that we have available to us in terms of the, let's say, dose-response information. You do realize that if you have a highly resolved measurement of, let's say, nanoparticle size distribution, a highly resolved measurement with some kind of a mobility spectrometer, you can convert the number distribution to surface or volume distribution. So for me, assuming the particles are spheres, so again you introduce some uncertainty there. So for me, the best possible scenario if you want to do workplace measurements is take a highly resolving size spectrometer, measure your size distributions there, convert them later on, if you will, to surface area or volume, and you have, within a certain range of uncertainty, you have a lot of information available about your aerosol. You can also tell whether it's multimodal, you can say whether there's more than one source, and what have you not. Okay. There are increasing capabilities now of making direct measurements of total surface area. In other words, you're throwing away some information. You're not getting size distribution information. You just say, I would like to know what the total surface area of my aerosol is within certain range of sizes. This already introduces some new issues because the surface area that you measure with aerosol instruments is not the geometrical surface area. It's not the surface area measured by BET. BET is an absorption technique which pretty much has access to the entire volume, a surface area of even a complex agglomerate, including interstices and pores and what have you not. The aerosol techniques measure accessible surface area, accessible by whatever technique you're using. That there's a factor between those. And people are at the moment, and if you look at scientific output and research papers in this field, people are fast and furiously working at the moment of establishing relationships between the different surface area measurements to be able to put at least an uncertainty bracket around that. There's a lot of confusion actually about it because people think surface is surface, but it's not. So again, you have probably something like a factor of 2 in surface area uncertainty. Also, what's emerging now more and more is you can get structural information about particles by direct measurement. You could always do that. You could do that 20 or 30 years ago with some pretty sophisticated laboratory techniques. It requires at least 2 independent measurements with 2 different techniques, but you can have that. But there are now new devices emerging based on established and even some new scientific concepts would allow you to give you some average, let's say a fractal dimension of the particles that you have, which is not bad because it gives you some knowledge about whether you really have to do with smooth spheres or something more agglomerated. There's other things that aerosol measurements can't really do very well or not very easily, which is equally important, and this is speciation. And again, here I wrote this explicitly here later on. I'm disregarding offline techniques for sampling and analysis. Of course, you can always collect particles on a filter or size fractionate them with an impactor, and then you can go away, and a couple of days later, you will know what was in the aerosol. But that's not very helpful for us. We'd like to know much more quickly. And there are techniques such as aerosol mass spectrometers. There's aerosol photoemission spectroscopy for certain surface species. And I'll say a few words about one of them because it's a really neat technique. But it's really hardly applicable or not applicable aside from cost to the workplace. But there are some new developments in this area as well because it has to be addressed. It's extremely important to know what you're actually facing. And I'll say a few more words about that in just a moment. And I will describe a little bit of what the nanodevice project is doing. Another real issue is to identify a specific nanoparticle source, let's say EMPs for the sake of discussion here, to identify them against the background aerosol. And the problem is there isn't any workplace, there is no room, there's no place where humans breathe, aside from a clean room in a chip manufacturing environment, which is totally free of background aerosols. You have plenty of them. So it really becomes a numbers game. Unless a source is sufficiently strong, you have considerable difficulties identifying it, and all those of you who are involved in monitoring emissions in the workplace know that. If you have 30,000 particles of regular background and you're trying to find an extra 10,000 somewhere from a source, that's not very easy. And as we know, most sources are relatively weak sources. There are a few cases where EMP or, let's say, human industrial activity really produces source strengths, such as transfilling, for example. If you take a powder and you move it into another container and things like that, or cleaning up a spill, indeed, there you can really have concentrations which are appreciable. But otherwise, in a reasonably controlled manufacturing environment, unless you have a big leak with an accidental release, you don't have much nanoparticles. So how are you going to find those small doses of particles? Well, again, the answer is speciation. If you could speciate your materials, if you could check what's in them, you could find these particles even if they were attached. And besides, I've added this at the very end, what information can aerosol measurements currently not easily provide? And I mean, they can't do it cheaply in many cases. Most devices, especially real-time measurement devices, are quite expensive, and they're also expensive because they require people who run them. Just a word about aerosol photoelectron emission spectroscopy, because I think this could be a pathway also for speciation in certain, let's say, in certain situations. APES is a technique where you illuminate your particles, you illuminate an aerosol flow with light, h nu, of a given frequency, and if you get the frequency right, you sort of ionize the particles. particles have a work function, and if you put the right amount of energy into them or a little more, you knock out electrons and the particles get charged. The electrons go away. And you can then, with a relatively straightforward technique, measure the average charge per particle. And that average charge per particle for a given irradiation is very much dependent on the surface. It's an extremely sensitive technique for surface analysis. You only need one type of measurement with fairly readily available aerosol instrumentation. This technique was actually used a couple decades ago for a while to look, for example, at adsorbed PHAs on diesel emissions. Because if you have diesel and you have diesel with an adsorbate of specific kinds, the work function of the adsorbate is so totally different that you can detect sub-monolayers of that stuff. And it would be really neat to use that, for example, in the aerosol state to look for, let's say, changes in the spectrum of charges, and to see whether you don't need to know what it is, you just need to look for a change, and then you can always go back and do other offline analysis to corroborate what you're finding. But it's a technique that's it's cool, really. The whole thing gives a lot more information about than just the work function of the materials, but I won't dwell on that at the moment because that would take me too far away from my talk. And the speciation, as I said before, is a way out of the dilemma of not being able to detect particles against the background. And I've already talked about this before, so I'm going to make it very short. Aerosol particles undergo a life cycle from the source, where they're emitted, some kind of device with a leak, let's say, and the receptor, the person who's in the lab or in the factory space and who breathes it. They're transported through space and time. They undergo changes by dilution, mixing with background aerosol. They collide with these background aerosol particles. They coagulate, as the aerosol people used to say, and they attach with 100% efficiency. They also get lost to the walls. Which mechanism prevails depends on the time, the source strength, the background concentration. But one thing can be generalized, and that is that the attachment kinetics of these freshly emitted nanoparticles to some pre-existing neutral ambient background particles is usually much faster, the attachment kinetics, than the other mechanisms. I've got a really neat movie for that. It's a very educational movie, and I'm going to show it to you again. There's two experiments that were done. In one experiment, there was a continuous source of nanoparticles, about 10 nanometer particles. In this case, platinum particles. They're easy to detect. And they were emitted into a space with or without background particles. And the background particles are in a totally different size range. They're in the size range where most background particles are. The most probable size is around a few hundred nanometers. Incidentally, also the size of cigarette particles. That's very often one of the big peaks in the spectrum. And again, the measurements were made. And I'll show you a couple of movies. And the first one starts by itself. You're emitting the source, and time passes. These are 15-minute intervals over 6 hours. And what you're seeing is you keep emitting a source. Of course, this source peak doesn't go away. But these particles begin to agglomerate. And as they're agglomerating, a secondary peak, an aging peak evolves, which are bigger particles. That's well known. Okay, this kind of aspect of aerosol dynamics is something that's about 100 years old, already this knowledge, and we know it. The really interesting movie is, if you do the same thing, you emit those nanoparticles into a space, and then you inject, artificially you inject, for 1 minute, a background aerosol. And here again, you have the source. It's already a little bit aged. And then here comes the background aerosol. And you see the background aerosol within one measurement interval of 4 minutes just mops up the entire concentration of nanoparticles. They're all gone because they just simply agglomerate with the background aerosols. Now, if you emit 10 nanometer particles and they attach to, let's say, a 200 or 300 nanometer particles, they're gone from the size spectrum. You won't see them anymore. But chemically, they're still there. Okay? So if you could do speciation, and of course with platinum it's not terribly hard to do. Platinum is, for example, also catalytically active, and you can then check whether you have your same catalytic activity. Well, yes, they're there, except they're no longer in the size spectrum where you've been looking at them. And this kind of highlights the difficulty, and aerosol science and aerosol measurement techniques are not going to a priori solve this problem for you because they can only measure what's there. But if the particles are no longer in the 10 nanometer range, you won't find them there. And you may conclude, well, the air is clean. I do hope that I haven't crashed my presentation, no, because I've already gotten stuck once with this movie here. Just to illustrate the kinetics of homogeneous versus binary coagulation. Binary means you're connecting big and small particles. Homogeneous means it's just the nanoparticles agglomerating among themselves. And this is the classical kinetics of homogeneous coagulation. You can see that it takes hours for this aerosol to change significantly. On the other hand, the binary coagulation is an exponential phenomenon, and it kicks in within minutes, typically, and within an hour you can see that the concentration has dropped orders of magnitude. Okay, so the kinetics are much faster of this binary coagulation. So let's draw an interim summary here. What do we need to know? With what accuracy? Well, we do need to know concentration, and we would like to know the concentration attached or unattached in terms of number or surface area. And you can already see that I can only answer this with a partially affirmative response. As long as they're independent, I can see them. As soon as they're attached, I really don't have an immediate and direct measurement technique available for you at the moment. So this is something that needs to be addressed, and it is. What would we like to know? Of course, we'd like to know something about the size distribution, because size distributions, as I said, are relevant for what's going into the lung. We also need to know something about structure. And the structure issue is actually emerging as a possibility, increasingly, where we can maybe do more in the future, also in the field. And well, how do we go about finding it out? That's what the last part of my talk here. We need to do some device development. And what I should mention, perhaps, and I'm not going to talk a lot about this because this is something that would easily take up another half hour. Aerosol people have developed all kinds of neat strategies for source identification. Okay? And those are not just plugging in an instrument and taking a reading of a size distribution, but they go about doing like differential measurements. They will go into different parts of a facility and they will check concentrations, and then they will look whether there are significant changes in the spectrum. And this does give them some, in medical terms you would say, differential diagnostic information about, well, there's something happening in that particular corner and it's probably nano, and there's probably a source here. And in the factory, you typically know more about it because you know what you're producing. You're not producing there's not just a black box, your device, you know what material and what size and you produce. So it is possible to do that, but it certainly is not a straightforward, simple measurement. It requires people who have a lot of experience in sort of teasing out this information. What I'm going to go and talk a little bit more is about device development. And, I can put in a plug here for our nano-device project. It's a 26-partner project, and there are a whole series of people actually developing new instrumentation. One of the objectives was to make it mobile, portable, personal, easy to use, affordable, all the kinds of things that aerosol people are not known to make their instruments like, because they, of course, love complex instruments. We love science. Scientists love complicated devices, and they know how to use them, but the people in the field don't like them. Of course, this requires some compromises required between accuracy and sensitivity and the detail of information, because you're giving this up for weight. It's nice if you have a device that you can sort of just take under your arm and walk into a lab, and that's actually what's happening there. And as I will try at least to get across, there are some new and some pretty innovative concepts. I don't even know how some of them work because they're still top secret, because people are still patenting this, but it's exciting, let's say. If you want to order it, there's one group of devices that give either total or size-specific concentration in real time, and NSM stands for number, surface area, or mass. Okay, there's all three involved in varying degrees of, let's say, industrialization. There are material-specific monitors in real time or in quasi-real time. I mean, real time is is what is real time. Real time does not necessarily mean you're going to get the information in a picosecond, like with some kind of laser. Real time also means if you have information within 5 or 10 or 15 minutes from the start of the measurement, that's also quasi-real time, and very often gives you time to react or to do differential diagnostic measurements. There's some samplers involved for collecting particles more or less fast with interesting techniques. And also there are some, the device development also involves pre-separator modules, which will cut out a certain part of the spectrum of size. And that's also not a bad idea, because we have devices, fairly simple devices already today, which will just give you a total concentration in some metric, surface area or number. But if you would like to know what is the size, what is the concentration, let's say, in a size range which is only specific to lung deposition, then you need to impose some kind of a pre-filter that will cut out that part. And so this is a module, if you will, that you can put in front of other devices. And so that's the idea of the device development. So here's a bit of statistics, 17 new devices, 14 have specific production plans or product plans. I do hope that some of them will really see the market, but there are companies involved, and as you know, companies do have a business objective, of course, and so I think some of those will really appear. And there are some feasibility studies as well. And I'm just going to go through this without spending too much time on the devices, just showing you the general, let's say, trend of what's happening. Concentration in real time by number. There are devices that will measure over very wide ranges, which is also useful because people who do exposure studies are not only interested in the nanoparticles, they want to know what the total exposure is very often. So it's good to have a few size classes in a very wide range. There are other devices which will give you this with additional information in a narrow range. These devices are very often based on electrical mobility concepts, but also on optical concepts. There is one which is a real total active surface area monitor, again, which is again a small box. You see, boxes have different sizes, of course. And in fact, there's a very neat futuristic development of using a cantilever device like you have in an atomic force microscope, and to be able to collect particles onto the cantilever and then to look at the change of the vibration frequency and to basically measure the mass of like a 100 nanometer particle. This is a bit further out. For sure, that's not going to hit the market next week, but if it works, we would actually have something which we don't have at all at the moment, which is sensitive, nanoparticle sensitive mass monitors, because at the moment we're mainly counting or measuring surface area. So if we could even get mass of a particle, that would be fantastic. The material-specific monitors, there are some that are really focusing on the carbon nanotubes, and when more information will become available, that will be really interesting to monitor their concentration, to be able to say this particle floating in air is actually a carbon nanotube. It's not just any particle floating around. That would be really interesting. And we ourselves are involved, among other things, in the catalytic aerosol monitors. In other words, something that will allow you to say what the concentration of catalytically active material in the air is. And we're also working on a fluorescence labeled surface species monitor, which is also one of the further out concepts. There's a whole wide range of things about offline particle analysis, which I won't go into too much at the moment because this is really going further away from my self-imposed limitations of saying I want to talk about real-time instruments. And in the end, yes, this continues with samplers for offline particle analysis. And in the end, of course, there are also the pre-separators that I have already mentioned, which will cut out certain ranges in the size distribution. For example, one will do 20 nanometers to 5 micrometers, the other one will do 5 to 400. So you have one which is for the gas exchange region, and the other one is for diffusional deposition in anterior nasal region. And if you spend enough time, you can tune these devices to give you specific size ranges. Since I've talked about catalysis, and since it's from our own lab, I have a few pictures for you to show. This is the catalytic activity aerosol monitor. What you actually do is you measure the concentration of catalytically active material by running in quasi-real time a small catalytic reaction with a specific model reaction. So you have to know what particles you're looking for. It's not just any particles, but if you know that you're suspecting there's platinum in the air, you could do something that will give you that kind of information. And just to show you the detection limits for palladium, nickel, platinum, and iron oxide, which are four really relatively classical catalysts. The palladium goes from 2 nanograms as a detection limit up to, for iron oxide, is the least sensitive to 28 micrograms. And the sampling time that you have, if you have a certain source, can range from seconds to minutes. So it's a quasi-real-time device. And the neat thing about it is that the detection limits and the sensitivity goes up in proportion to the degree of, let us say, not danger, but not dangerousness of the particles, because the more catalytically active something is, the more risk it presumably poses, and therefore also the more sensitive your technique becomes. I wouldn't be an aerosol scientist if I weren't also putting in a plug for my own science. And what I wanted to do at the very closing of this talk is to say that aerosol science can do a lot more for you in terms of the bigger issue of assessing risk and assessing, for example, biological effects. Aerosol science has a fantastic set of tools for designing particles of all kinds of degrees of complexity. Those are my favorite ones. That's something we make. These are nanoparticles which are coated with dots of a noble metal, and you can tune the dot size in steps of 1/10 of a nanometer, and you can look at the catalytic effects. And just to show you that for palladium, the catalytic effect is a very, very strong size of the particle size. So this is a way, and in fact, we can characterize and design this material with measurements. So actually, you have extremely well-defined particles to start out with, and that, of course, brings me to another issue. I'm a big proponent of the air-liquid interface exposure systems, because this would be the best way to take these particles in the most unadulterated way and bring them in contact with a biological system. And just to show you also what kind of information you can tease out of a catalyst in real time, this is the comparison of structural change versus catalytic activity for a platinum catalyst. And what we're doing is we have agglomerates which start out with a few nanometers in primary particle size, and then we gradually change the temperature. We restructure the particles, and we also gradually change the primary particle size thermally. And we look, on the one hand, at the exposed surface area, because as the agglomerate becomes more and more compact, more and more of the surface area becomes internalized and therefore is not available for the reaction anymore. We change the primary particle size, and we want to, at the same time, look at the catalytic activity. And the blue curve is actually the catalytic activity, which you see the catalytic activity begins to change, although the exposed surface area hasn't changed at all. Okay? And all of this is measured in real time. And we know from aerosol photoemission spectroscopy that chemically we haven't changed the surface of those platinum particles at all. The emission function is the same. The surface is absolutely pure. So the only conclusion you're left with is that it's the primary particle size which is moving gradually from 5 to 6 to 7 to 17 nanometers, and this is changing their catalytic activity. And if you have those particles available, you could, of course, do also some fantastic studies of their biological functionality. And with that, I would like to thank you very much for your interest, and I hope I haven't taken you too far away from the main of my presentation, and I'm open, of course, for questions and discussion. Thank you.
Thank you very much, Dr. Kasper. Fascinating talk, beautifully presented. Do we have any questions for Dr. Kasper? Okay. I thought she was good.
Could you elaborate a little bit about the what you called the direct measurements of the total surface area and the factor 2 of error? You were talking about BAT.
Yes, these are two different issues, of course. Direct measurement means you have a device which gives you measures the amount of particles you have present in terms of its surface area. And these techniques most of these techniques which are currently in use do this on an electrical basis. In other words, they measure the degree of absorption of electrically charged ions from the surrounding air. So it's actually, if you will, in a physical-chemical sense, it's an absorption process, okay, but a very specific one. And the neat aspect about the absorption of electrical charge is that you can immediately measure the level of the charge of the particles individually, so to speak, right afterwards, after this attachment process, and thereby you gain information about the surface area. In fact, if you do it right, you can even get information about structure also. And as far as the factor of 2 is concerned, if you take 2 or 3 different aerosol instrumentation devices, each based on a different operating principle, say one based on optical diameter and the other one measuring electrical mobility, and perhaps even electrical mobility based on different ways of charging the particles electrically, each one, and for the same aerosol, will give you a somewhat different size information, which has to do with the fact that each particle, each of these devices was calibrated in some form. None of these, or almost none of these particles, are absolute devices. You need to put something of a known, some of them are, okay, but and usually you have to put in particles of a known size. And so you use calibration aerosols, and that's polystyrene latex for optical particle counters, for example. And the result is that you get differences in size. Okay. These differences in size can amount to 1 or 2 size classes, but in my experience, a factor of 2 will typically cover that. Okay. You can do better if you know what you're doing, of course. I was just sort of drawing an upper limit for someone who takes a device, just bought it happily, takes it to the lab and says, okay, I'm going to turn it on. With what kind of a degree of reliability can I really say that this was exactly the size? Okay, if I get size information, then I would say a factor of 2 isn't too bad. Okay.
So if I understand correctly, what you mentioned about the surface area being measured by what you described is mostly how electrical charges are being distributed at the surface.
Not distributed on the surface. How many are absorbed per particle?
Okay, but this is far away from a good information about any surface reactivity from a chemical point of view.
No, but it's a measure for your surface area. And if reactivity is proportional to your surface area, then basically if you have one aerosol which has twice the surface area of the other, that will tell you something about which one is likely to be more reactive. Okay. Of course it doesn't provide you with the chemical. For that you need a technique for looking for reactivity. But if you're looking for reactivity in the liquid phase, anyhow, how can an aerosol measurement provide you with this information? If you look for catalytic activity, it's the same. I mean, nobody guarantees you that the catalytic activity of an airborne particle remains the same. In fact, that's one of the things we'd like to find out now, okay, is whether that changes. I've talked about how to describe workplace aerosols reliably, okay. I've not tried to describe the reactivity or the consequences of this measurement in assessing something in a totally different environment. In fact, that's what this is all about. That's why we're here, okay? We're trying to build a bridge, and I hope that I was able to contribute to sort of putting the two sides of the bridge together a little bit.
I think we have time for one more question.
Hi, first of all, greetings from my thesis advisor, Dr. Mark Hoover, right here at your institute, as we speak, in fact. He's going to be on Skype here tomorrow. Okay. You said, you gave a beautifully elegant presentation, and I thank you very much. And you said two things that struck me as almost radical, if I understood them, and I may have misunderstood. One is that there's no place without some kind of background aerosols except a clean room, and so it seems like we're almost always playing some kind of a numbers game. Yep. That's, I guess, not good. And if I understood BDG, the movie, do we in some way end up rewriting regulations because of what the movie has taught us? And the question I have for you, therefore, is how would one think about the place of these very important new ways of understanding aerosols in a regulatory context? And I don't expect you to answer the whole thing right away, but I do want to sort of start for the ship that will sail these seas. Thank you.
Well, indeed, and I don't know whether I still have a microphone, but that's okay. It's back. Indeed, there is always an aerosol background wherever you are, okay? Because there are multiple sources, and if you measure aerosol in this room, for example, here, you'll find a fairly characteristic size distribution, which has a maximum somewhere around, very often around a few hundred nanometers, and unless you have some strong nanoparticle sources, it'll sort of fall off a little bit towards the very small end, and there may be, of course, a coarse mode after that. This is known since Ken Whitby. That's nothing new. And you're trying to measure a needle in a haystack very often when you're looking for the nanoparticles, because naively speaking, you're looking for them only with one single approach and one dimension, that is numbers in a certain size range. So anything that's within, naively speaking, 10 and 100 nanometers is a nanoparticle, and we need to worry about it. But who tells you it's not just an innocuous ambient particle? Okay? And that's why I think we need to put a lot of emphasis on speciation. We need to look at what is it in real time. And people can do that already. I mean, you can buy fantastic aerosol mass specs, which are sort of half my office in size and which cost a few $100,000 or euros. But the problem is you can't ask anybody to really buy them and put them into the workplace for routine measurements. But we need to make them cheaper, more effective, maybe more specific. But I think that's the way to go. Because if you can tell that, let's say in the 300 nanometer range, which is the most prevalent in the size distribution, you do have, to stick with my own example, you do have platinum there, okay? And in fact, if you go to the electron microscope, you might find some of that platinum if you're lucky, and it's still 10 nanometers or 20 nanometers, it's attached. But chemically, reactively, it's still there. In fact, that's what we've already begun to show, that the chemical reactivity of those particles when they're attached, of course, a supported catalyst is not exactly the same as the unsupported, but it's pretty much the same thing. Okay? So you really need to worry about where are the particles also, and they could be in the 10 micrometer range, but they're still catalytically active.
Okay. Well, thank you for that question and also for that answer. If we get through our program on time, I'm hoping to save a bit of time so we can come back to these issues, because I have some experience from other direction in industry where you get this balance of real-time measurements versus the need for regulation and also validation. So with that, I must thank Dr. Kasper for his excellent presentation, and on your behalf, present him with a little gift. Yes, you will.